The information stored in DNA is used to make the protein machinery responsible for controlling all of the functions of the cell, from movement to cell division. The process of constructing these proteins begins with the production of messenger RNA, which in turn is used as a template for protein synthesis. In short, DNA is transcribed into RNA. There are three types of RNA. Messenger RNA, or mRNA, is used as a template for protein synthesis. Ribosomal RNA, rRNA, forms important components of the ribosomes, which are responsible for translating the mRNA template into chains of amino acids or proteins. Transfer RNAs, tRNA, carry amino acids to the ribosome. RNA is similar to DNA. However, it consists of nucleotides containing ribose and is single-stranded. Transcription starts with the binding of an enzyme, RNA polymerase, to a region of DNA known as the promoter. Like DNA synthesis, the synthesis of new RNA strands proceed in the 5' to 3' direction, with the addition of new nucleotides to the 3' hydroxyl end of the RNA chain. DNA is used as a template to build the strand of RNA. Complementary bases hydrogen bond with the nucleotides of the DNA. The base pairing is similar to DNA. However, instead of thymine, RNA contains uracil, which base pairs with adenine. The process stops when the RNA polymerase reaches a sequence of DNA called the terminator. Transcription begins with the binding of the RNA polymerase to the promoter region. The polymerase begins to temporarily unwind the DNA double helix. This allows the RNA nucleotides to base pair with the DNA template. RNA polymerase then catalyzes the formation of the sugar phosphate bonds that hold together the backbone of the new nucleic acid. As RNA polymerase moves along the DNA, the transcribed DNA rewinds. Once RNA polymerase reaches the terminator sequence, it releases both the DNA and the messenger RNA. In eukaryotic cells, the RNA is then processed before being used to synthesize proteins. Eukaryotic genes contain regions known as exons and introns. Introns do not code for protein. These introns must be removed from the mRNA transcript before proteins are synthesized. This process is known as splicing and allows cells to generate modular pieces of RNA which can be spliced together in many different combinations. Once the mRNA is processed, it then leaves the nucleus and can be translated by ribosomes in the cytoplasm. Prokaryotic mRNA is not processed before being used for protein synthesis. In addition, prokaryotic transcription occurs in the cytoplasm because there is no nucleus. This allows ribosomes, the structures responsible for protein synthesis, to bind to an mRNA molecule and use it to build proteins before it is even completely synthesized. In addition, multiple ribosomes can bind to a single piece of RNA at one time. This is known as a polyribosome. It allows the cell to synthesize large numbers of proteins in a very quick fashion. In translation, the sequence of nucleotides found in the mRNA is used to construct chains of amino acids, which make up proteins. This process occurs at the ribosomes. Groups of 3 nucleotides make up codons, which correspond to specific amino acids. Translation begins at the start codon, AUG, which encodes the amino acid methionine. Amino acids are then added in sequence to that first methionine. Translation stops when the ribosome encounters a stop codon, or non-sense codon, which does not correspond to an amino acid, but instead tells the ribosome to release the mRNA template. As you can see in this table, there are 64 sense codons, which encode 20 different amino acids. A single amino acid, such as valine, can be encoded by several different codons. Here, valine is encoded by 4 different groups of 3 nucleotides, or 4 different codons. For this reason, the genetic code is called degenerate. This allows for small changes in the DNA sequence without affecting protein structure. tRNAs, which carry amino acids to the ribosome, have complementary sequences called anticodons. For example, a tRNA carrying valine would have an anticodon with the sequence CAA. Translation converts the message stored in mRNA into protein. The information stored in the language of nucleotides is translated into the language of amino acids. It begins at the site of the start codon, AUG. At this point, the subunits that make up the ribosome come together, along with the tRNA carrying methionine and the anticodon UAC. Once the ribosome is completely assembled, the start codon is lined up with the P site of the ribosome, where the complementary tRNA carrying methionine is in position. The next group of 3 nucleotides determines the next amino acid in the sequence. Here, UUA encodes for leucine. The tRNA carrying leucine and the AAU anticodon moves into position at the A site of the ribosome. At this point, a peptide bond between methionine and leucine is formed. The ribosome continues down the mRNA. The now empty tRNA that carried methionine moves into the E, or exit site, of the ribosome. The second tRNA, still attached to leucine, moves into the P site, and a third tRNA, an amino acid, move into position at the A site. This process continues with the release of the first tRNA and the formation of another peptide bond. The ribosome moves along the mRNA chain, adding new amino acids to the growing polypeptide. Eventually, the ribosome encounters a stop codon, which does not have a corresponding anticodon on a tRNA. This signals the release of the mRNA, the final tRNA, the ribosomal subunits, and the new protein. The ribosome moves along the mRNA from 5' to 3'. As the ribosome proceeds down the mRNA chain, the start codon is exposed and additional ribosomes can bind, allowing multiple proteins to be built using a single mRNA transcript. In our next lecture, I will discuss the process of regulating the production of new proteins as needed by the cell.